today I'm delighted to be introducing you to Luíseach Eoin, or Doctor, I should say, who's coming all the way from London to be with us here tonight. And she's Irish originally, but seems to have quite an affinity to Southern England, I would say.
Oh, I'd never say that.
No? Okay. So she seems to have gotten there and not left very often, but I think...
I have an English husband, so I have to be nice.
Okay, no option. So she has studied at some of the UK's most prestigious universities. She did her undergraduate in archaeology and anthropology at the University of Cambridge before moving on for her master's in Paleolithic archaeology and palaeoanthropology at the University College London. Then for her PhD, she made the move to the University of Oxford, where she started to research hunter-gatherer archaeology and heritage conservation. And for her PhD, she spent a lot of time doing fieldwork in Southern Africa as well. So well-traveled in that regard. Then after completing her PhD, she actually transitioned to the world of academic publishing. So I think she had more sense than some of the rest of us and realized trying to be an academic isn't always the smartest career choice. So first she was an editor at Nature Plants and then quickly moved to Nature, Ecology and Evolution. So again, she's primarily based out of her London office. And as an editor of some of the world's most prestigious journals, she actually gets advanced access to the latest scientific advances. She gets to see what's happening before us, and she gets to shepherd that research through from its early phases all the way through to completion. So I think you get a good insight into what's going on and also have kind of a bit of a say and can act as a bit of a guide as to what is considered to be rigorous science and what's not quite there yet. So she's, as an editor, handles a wide range of topics, and she's told me it ventures far from what she originally trained in. But generally, she's dealt with things like animal communication, also things like extraterrestrial ecology. So I say she's getting further and further removed from the past. But her main focus of articles that she handles is really anything got to do with fossils, particularly using fossils to understand how ancient organisms worked, how they interact with each other, how they interacted with their environments as well. And this also includes that overabundant animal humans. So a lot of our focus is on human research as well. And I'm delighted to say that she's going to take us on a tour today through the absolutely latest developments. Like I said, she gets advanced access to all these things of human evolution. And along the way, she's going to give us some insights into the life of an academic editor as well. So Dr. Eoin's talk is titled Bones, Stones, and DNA, a nature editor's view of human evolution. So welcome, Luíseach. Look forward to your talk.
Thank you so much for having me. It's really great that Dave gave me that introduction because actually this talk started with a bit of a crisis of confidence. I was thinking, I'm not a researcher anymore. What right do I have to come and talk to you about human evolution, which I think is one of the most exciting fields that there is today. And while I was going through this crisis of confidence, I came up with a second title. Why am I here? What brings me here to talk to you? So I actually thought that was a good sort of thing to structure the talk. So I'm going to break it into two halves. And the first half is going to be why I'm here right now. How did I come as a nature editor to be talking to you about this topic and tell you a bit about what my day-to-day job is and why I think it's an important one. And then the second part of the talk is still going to be why I'm here, but it's kind of why we're all here. It's why we're all here at the moment, how our human evolution has led to this, and some of the exciting developments that are happening today. And it's a really huge field, so there's not time to talk about everything, but I'll just talk about a few things. So without further ado, this is me. So on the top left up there, you can see me doing my PhD fieldwork in Southern Africa, where I was looking at what plants that hunter-gatherers ate something like 40,000 years ago to the present day, nearly. And then I've ended up on the bottom right, that's my desk at work, where I spend my days reading scientific manuscripts, deciding what ones to send out to peer review to make sure that they're technically scrutinized before being published in Nature, which is one of the most prestigious scientific journals there is today. And I should also mention, because I'm in North America, that other scientific journals are available. There's a really good one called Science, which some of you may be more familiar with. And I also wanted to say that the closest I ever came as a researcher to publishing my own research in one of these journals is when I featured in a photograph of an excavation I was working on. So if you look up there at the small n in human and then the big M in migrations, that's me bending over it. So that was a big day. It was really exciting. So as a researcher, I'm up there doing my field work. I'm there digging in North Africa as well. I kept being distracted. I would get my attention grabbed by all of these really cool, exciting topics that were happening, and I would get distracted from the job I was supposed to be doing. And if you have been a researcher, if you've done academic research, you'll know that there's sort of a pressure to specialize, and that this kind of tunnel vision is really essential to being a good academic. And only later in your career do you get to broaden out again and think about bigger questions. If that's okay. And I just kept getting distracted. I would get distracted by all of the cool different areas of research that were happening. So when I came to finishing my PhD, I had this question: am I going to stay working on this little postage stamp style area of archaeology, or is there a chance that I could get to see a bigger picture? And at that time, I saw that Nature Plants, which I'll talk a little bit about the different Nature journals later, but they were hiring an editor. And I thought, what does that mean? I knew that Nature published some of the most exciting research in the world, and that, if your research was published in Nature, that it must be really, really good, that it must be, really fundamentally important. But how that process happened, how someone's research went from something that they found in the lab or in the field to coming out on paper in a magazine, I didn't know how that worked. So I went to it kind of blind. But 7 years later, I'm still doing it. And I get this really exciting chance to help this process happen, help the research turn into a published paper. And it's taken me all over the world. So on the bottom left, I'm in Olduvai Gorge, which is in the part of Olduvai Gorge that I'm in is in Tanzania. And some of you may have heard of it. It's where some of the most exciting discoveries in human evolution have been made. And the funny thing is, you'd think I was working in Southern Africa, maybe I would have gotten to go there as part of my job if I'd stayed in academia. But actually, it was such a different field, even within that sort of small area of human evolution. I might never have gotten to go there. But I got to go last year as part of my job now when I travel all over the world talking to scientists about their work. Is that coming from me? I'm not sure what's... Oh, sorry, I'll try and stop doing that. So yes, that's why I'm here today. So first of all, I'm going to talk to you a bit more about Nature. So Nature was founded in 1869 by a man called Alexander Macmillan, and it comes about at the end of maybe the most exciting decade for science in the 19th century. So Charles Darwin published On the Origin of Species in 1859. You had things like the discovery of Neanderthals and Cro-Magnon man, also in the 1860s. A man called Charles Lyell linked Darwin's ideas about evolution to the rock record, to stratigraphy. So we could tell that the world was really old and that there had been transitions over time between different kinds of creatures. And so all of these ideas are coming out at this time. Things like railroads foster the discoveries and how they could be transmitted between areas. But at this time, Alexander Macmillan was having these things that he called tobacco parliaments. And he'd have these meetings every week in his home in Cambridge. And some of these learned men would come together, and they were usually men. There were some women involved, but it was altogether a pretty elite kind of meeting. And he sort of went from these tobacco parliaments, which are very much a continuation of, if you've heard about the sort of 17th century coffee house culture, where people like Isaac Newton would have met, come together to drink coffee and talk about these really exciting discoveries. Macmillan's kind of tobacco parliaments were very much a continuation of this. But then Macmillan does something interesting, which is that he sort of formalizes this into a print journal, publishing every week. So while science is still sort of advancing by these elite, usually men, presenting to a community of their peers in person, kind of doing these oral presentations, Macmillan starts writing reports about them. So in Nature, you will get the dissemination of these scientific ideas being published so that it can be spread around the world. Now, this kind of format of reporting on what people were talking about remained the format for maybe the first 30, 40 years at Nature. And it took 30 years to stop making a loss, which is maybe somehow related. And you can see that some of the publications that things they were publishing in those days were a bit odd. So on the left, we have a carnivorous goose. Someone thought that was interesting enough to report upon. And it is just worth noting that what I said about sort of these were elite people publishing this. Argyle, who's the author there, he's a duke. a royal duke who, you know, thought that his observations about this goose that apparently ate meat were worth the scientific community knowing about. And then on the right here, you have a squirrel that can sneeze on command. And one of the things I really like about this is that if you're a scientist, you know that sample size is everything. It's really hard just to generalize from one sample. So we go from the carnivorous goose is n equals 1, but of squirrels, n equals 2. So that's a really exciting development there. So this is the kind of thing that they start publishing. And then over the years, we see nature progress. So it's now been over 150 years of nature. And in that time, some of the most exciting scientific discoveries, particularly of the 20th century, have appeared in our pages. So we have the discovery of the neutron, nuclear fission. We have the structure of DNA. We have the Homo habilis from Olduvai Gorge, the place that I showed in one of the first slides. And then we have all the way up to the sequencing of the human genome in 2001, and so many other discoveries as well. And Science is another prestigious journal that is also publishing discoveries like these. And gradually over time, we move from that forum of people presenting in person and maybe taking questions on the day from people who were there physically, but then over time, peer review. So as things are being published in a written down format, they're being scrutinized by technical committees of peers, other scientists working in those fields who can certify their technical accuracy. And this has become hugely important to publication in Nature. So it is worth pointing out that the structure of DNA, even though it's published in 1953, which you'd think is quite late in the sort of development of the scientific publishing method, actually it wasn't peer reviewed because it was regarded as so self-evidently perfect that it didn't need it. And that is something that would never happen today. There is no discovery that's so self-evidently perfect that it doesn't need technical scrutiny to make sure that you can trust in the accuracy of the results. So these are some of our successes. These are some of the things we got wrong. So up on the left, does anyone know what that is on the top left there? It's the Krebs cycle, the citric acid cycle. So it describes the means by which organisms that respire do it. And Hans Krebs submitted that to Nature in 1937, and it was rejected. He later won a Nobel Prize for it. Nobel Prizes, rejecting research that led to Nobel Prizes is, I don't want to call it a specialty, but it does occasionally happen. So you may be aware that recently, a couple of weeks ago, the Nobel Prize in Physiology or Medicine this year was awarded for discoveries relating to mRNA vaccines. Now, this is a key publication that's on the Nobel Prize website for that discovery, and you'll see that it's published in a journal called Immunity there in the italics. It was rejected by both Nature and Science. And then, apart from the things that we didn't publish, there are also some things that we did and we shouldn't have. This is Piltdown Man, which again, some of you may have heard of. And there was huge excitement about Piltdown Man when it was published in Nature in, I think, 1912. And that was because it was regarded as the missing link. We'd finally found proof of the link between apes and humans. Now, as some of you may know, we don't talk about that anymore. There is no missing link species. There's no perfect link between apes and humans. We know they're related through many other means, such as fossils and molecular techniques. But Piltdown Man was a hoax. So the reason it looked so perfect is that it had a human cranium, the top part of the skull, and it had an orangutan jaw. And these two things have been put together to make this perfect fake. And yet it's still past publication in Nature, possibly because this really important process, peer review, was still in its infancy at the time. I'm not sure. I'm also not sure why there's a goose in this photo, and I don't know if it ate meat. So there's that. So where are we with Nature today? So the first thing, and as I mentioned at the beginning, I was going to talk about how Nature isn't alone. So at the moment, I think there are something like 11 million scientific papers indexed on the net. That these are all of those that have been archived through time. Something like 80 to 90% of all of the scientists that have ever been alive are working today. They produce a lot of content, and a lot of really, really good content, it should be said. So Nature, a few years ago, started subsidiary journals in different fields. So Nature publishes maybe the most exciting research across all topics, but then we have these different communities. So Nature Genetics will publish the most exciting discoveries in genetics. Nature reviews cancer, will review cancer manuscripts, oncology. And the journal that I primarily work at, Nature, Ecology and Evolution, that's what EcoEvo is, will publish the most exciting content in ecology and evolution. So our rationale for what we publish is that in Nature, if you see a paper there, even if you don't understand the content of the paper, and I don't understand a lot of the content that's published in Nature because it's way outside my expertise or my learning, you should kind of get a sense from the way that it's talked about, the way it's presented, why it's there. So if you're a physicist, you should understand why a biology paper is being published there. Otherwise, why is it? And in these kind of sub-communities, we think that the important research, that bar we're looking for, is within that community. So if we publish an ecology paper, an evolutionary biologist needs to know why that's important. So it's within that field, the really exciting thing. And while we're still talking about the scientific paper that's being technically peer-reviewed as the medium by which science progresses, these days, you're much more likely to read about it in your inbox, your email inbox, than you are in printed form. A lot of these journals, most journals today, are actually online only, rather than being published in print. And sometimes you hear about things like impact factor. I don't know if any of you have heard about that, but it's the number of times that a paper has been cited, that it's been used again, and the work attributed to the scientists that published it. And that is a metric to show imperfectly how impactful a paper is. But actually these days, what we're looking for is work that's impactful, maybe in a more societal sense, that it's making a real difference to society. So that Nobel Prize winning work that we didn't publish back in 2005, but the fact that the Nobel Prize is being awarded for those mRNA vaccines, the kind of ones that were so fundamental in COVID vaccinations, shows you that kind of societal impact. We're also looking for work that's rigorous. So that's the peer review, the technical certification of correctness that I'm talking about. And also that it's reproducible, which means that the results that a scientist publishes in Nature, someone else can come along, take that data, and then replicate those results. They get the same results again. It's not that it's hocus pocus that someone has somehow manipulated something to make it publishable. So those are sort of the values that we're looking for today. And that's where my job comes in. So it's my job to make sure that those standards are upheld. So I spend my day deciding what papers we should publish. So I read a lot of manuscripts in lots of different topics, which is fabulous for my sort of magpie-like attraction to new topics. I get to go down a wormhole of excitement every time I read about lizards or extraterrestrial ecology, which still totally blows my mind. Even though we don't actually know if there is life on other planets, people are trying to work out the signs by how we would be able to tell if there was, which is incredible. So we decide by reading the papers, by reading the literature that they cite, the ones that the previous research that they mention, where it falls in the field and why it might matter. And then once we have a hunch that that might matter, then we send it out to peer review. So the important thing to know is that I am not a technical expert in most of the stuff that we publish. Sure, I tend to have a pretty good handle on some of the topics because maybe I did my PhD on those things, or I happen to have read quite a lot about them. But we publish so much and in so many different areas that I couldn't possibly have that technical expertise for each one of them. But I don't have to, because it's my job to find experts who are reliable, who do know about those things, and to make sure that we have enough of a panel assembled that they can check whether the claims made in some kind of discovery are accurate, whether we have evidence to support them. And sometimes that takes a while. You don't necessarily get the perfect version of a scientific paper in the first go. It can take many rounds of revision and then re-review before you get something that's publishable in any journal, let alone Nature. So I keep coordinating that. So paper comes back from review and they say, well, if you did this experiment, maybe you'd have some better evidence basis from which to argue that thing that you want to present. And so we might send to the authors, please, can you go back and try that, see if it works? And they go back and try it and comes back. They've got the results that they wanted. The reviewers will go through it again and say, yeah, okay, we're happy with this now. We think insofar as is reasonable, you have an accurate evidence base for making the claims that you have. And so this is sort of the most important aspect of my job. And one of the key things is that we try and stay involved with the research community so that we can make those judgments about what might be interesting. So I'm really lucky that I get to come to places like the Whitney lab to find out all the really exciting research that they're doing, so I can better understand the papers that were sent, so I can help evaluate them and then send them out to those experts. And one of the really important things about my job is that I'm neutral. The idea is that because I don't have any skin in the game, I don't have a vested interest in seeing any particular kind of paper or area published, other than that it's interesting. So I can try and be an objective witness to how this happens and to make sure the process is really fair. And one of the things that I can try and do is to make sure that the research that we publish reflects the diversity of society, which is hugely important. And we know that nature had a reasonably progressive attitude to female scientists early on. So I was trawling the Nature Archive for things like the carnivorous goose and those squirrels, which is just really, really fun to go and find out about these early papers that were published. And I was trying to find out who was the first woman scientist to be published in Nature. And I couldn't find it. I don't know who it was. But I do know that in 1872, Mary Somerville, who's a mathematician, was regarded as important enough by her peers that she warranted an obituary published in Nature. And that's a really big deal. These days, Nature tends to publish obituaries of Nobel Prize winners. At Nature, Ecology, Evolution, we try to be a bit more egalitarian than that, but it's definitely a selective process. But so Mary Somerville was regarded as important enough as a contributor to science at that point that she warranted this obituary. So we have this sort of reasonable advancement of fairly elite white women scientists at this time. But over time, we know there are a lot more barriers to people from diverse backgrounds in STEM, that people of color, people from low-income countries, LGBTQ plus individuals, they all face a lot of barriers to science and being able to do their research. But what we also know is that science really benefits from diversity. So research has found that diverse labs are more productive. They publish more, they produce more research. We also know that who you are affects the actual development of science, the things that we find out. So for a long time, amazingly, it was only thought that male birds sang to communicate. It was only with the gradual inclusion of women researchers in birdsong research that we found out that actually, well, female birds do sing, because of a particular interest that those women had. We also know that things like where you come from and where you're able to research, where you have funding to research, can affect things like the fossil record. So we know that, for example, if you were a scientist from the UK, you might have, for example, a historic link or funding to go and research in Morocco. And that will influence the kinds of discoveries that are made in Morocco. So we know more about dinosaurs published from this area of the world because they happen to have money to research them than in areas of the world where people lack that kind of funding, which is really interesting. So one of my jobs is to try and ensure that we have diverse people doing science, evaluating science, and advancing it. So I've got a bit of a bridge here to the next part of this, to the next part of the talk, where I'm going to actually talk about some of those human evolutionary discoveries now that I've hopefully explained why I'm here and why I think it's important to look at these in sort of an objective light. But so first of all, I want to ask, has anyone seen a Netflix documentary called Cave of Bones? I know two people up front have. Anyone come across it? Okay, well, maybe you'll want to go and watch it after I talk about this. So Cave of Bones, this is the Cave of Bones. This is Rising Star Cave or Dinaledi Cave in Southern Africa. And then it actually happens to be really close to where I did my PhD research, although it wasn't related. And that's how I know that Naledi means star in the locally spoken Sesotho language. And that's kind of fitting because Homo naledi, who's this guy, is a real star. I've got a little fact file about Homo naledi here, the kind of dates that he was alive, or they were around, and some of the things that they might have done. I'll go into that in a second. Homo naledi has been recently the subject of this Netflix documentary, because Homo naledi has been claimed to do some really interesting things. Despite being a pretty small-brained hominin, that means someone of the genus Homo, one of our fairly close ancestors, we think. Sorry, fairly close relatives, I should say. Homo naledi has been claimed to have had control of fire, maybe had rock art, that means cave paintings, Maybe buried their dead. Maybe they had, a belief in some kind of afterlife or respect for human remains. Homo naledi has also recently been into space. So recently on a SpaceX trip to orbit the Earth, part of Homo naledi was sent into space. And a lot of people were kind of questioning this decision. And one of the things about Homo naledi is that in addition to being a bit of a superstar itself, its discoverer is a media star. Its discoverer is a man called Lieberger, who has done amazing things to advance the cause of science and paleoanthropology and South African fossil discoveries in the world. He also has done things a bit differently. So this is a really exciting discovery. All of the things relating to Homo naledi are, but very few of them have been published in Nature journals. Instead, Lieberger has gone for a journal called eLife, which has a really radically different publishing model from how we do things at Nature. So sometimes in our pursuit of science that we say is impactful, people say that's a form of gatekeeping, that we're making a selective choice about what science is and isn't important. And that's a valid concern, because I think the way that we do things, that selectivity is both it can be an advantage, but it can also be a disadvantage, because we're sort of gatekeeping this. And so eLife started with a radically different idea, which was not to make those kind of selective decisions, that they would publish what they call sound science. So good science that was peer-reviewed and certified, but correct by the kinds of technical peer reviewers that I talked about before. But they wouldn't go into things like impact. And they have some professional editors like me. They also have a lot of academic editors. And the difference between a professional editor and an academic editor is that an academic editor is still a practicing researcher. And a lot of journals have academic editors, and it can be a really, really good way of running a journal. And you do get, technical expertise, but you also don't necessarily get that kind of objectivity that I was talking about, that I hope I bring to the way we do things at Nature. Sorry, I'm very jet lagged, so I'm hoping I'm keeping the run of things. So this is all very well. A lot of this is very laudable. They publish some really great science, and I would have been delighted had the early papers on Homo naledi come to us at Nature. It would have been a fantastic privilege to publish them. eLife, though, with its goal of constantly pushing the envelope when it comes to the way science publication works, in trying to innovate, which is hugely laudable practice, they've recently changed still further. So where they used to still make a sort of selective decision when it came to accepting or rejecting a paper, now eLife works as a bit more like what we call a preprint server. So preprints are pre-peer-reviewed research usually. And what they do was they will put these up on their website. They'll put these papers, fully written scientific manuscripts, and then they will subject them to peer review, but they don't make a decision on whether to accept or reject them for publication. So when I mentioned some of the sort of controversial findings or controversial claims that have been made for Homo naledi, like deliberate burial of the dead or this rock engravings, those are controversial because there's a saying in all of science that extraordinary claims need extraordinary evidence. Having a really small-brained hominin that's really old do these things is a bit controversial, because as far as we know, there's a very limited club of hominin species, human relatives, that have done this for sure, we know. So our species, Homo sapiens, has done all of these things for tens of thousands of years in some cases, hundreds of thousands of years in others. Neanderthals, it seems like, have a reasonable evidence basis from which to say that they did some, maybe not all of these behaviors. I'll come back to that a bit later. But having a small-brained individual as old as Homo naledi engage in these behaviors was pretty radical. And unfortunately, when the reviews came back on these papers, the reviewers, which, these again, they're technical experts in the field, who scrutinized them thoroughly, they found that they don't have enough evidence at the moment to support these claims. It might be possible that they might find future evidence in the future, future evidence that might more securely back this up, but at the moment they haven't got it. But so what eLife do is they make a pretty high level assessment. So I think if you read this, you need to have a fairly technical grasp of the claims here and the field here in order to evaluate what this statement means. And what they're saying is very neutrally, the evidence is incomplete. But kind of the problem with this model is that there's no, put it in crude terms, seal of approval on the end product. So at Nature, we believe that our name stands for this kind of integrity of evidence, that the claims made in our journal are almost always, obviously, there can be human errors, there can be errors in any system. They are approved, they are technically watertight, they are rigorous, they're well done. And so if there's something published in Nature, we really, really hope that you can trust it. And so when stories are picked up in the media or consumed by the public, that you can trust them, because if you've seen the name of the scientific journal on them, we like to think that that means that there is a mark of approval on it. With eLife, this model particularly, you don't get that seal of approval. Now, the problem with the system is that it can be gamed. That if you have a study that is maybe making quite a splashy claim and you present this to this system, that it doesn't get that kind of yay or nay approach to it. Whereas if you're publishing something that's a bit more neutral, that maybe doesn't speculate quite so much, that maybe this system is actually fine. But the problem is that human evolution, human origins, is a hugely exciting, it's a topic that people want to know about. I'm guessing that maybe a lot of you came along this evening because you're interested in where you come from. You're interested in your ancestry, the story of where we came from. It has huge political currency. People like to make, claims about their origins, or it's really important where their ancestry is. There's a lot riding on it. And to me, that means that it's really, really important to make sure that it's trusted, sound evidence, what is being put out there by the media. So when you have Netflix documentaries that are based on research that isn't yet kind of technically certified to be in support of its claims, that's kind of dangerous because people can then use it to push a narrative. So I'm hoping that this kind of section has told you a bit about why I think that Nature is a good thing on the whole, and that it gives us, that if you read science published in our journal, that you can trust it, is basically what I'm trying to say. It's not to say that you can't trust research published in other places, but that we believe that this method, this sort of seal of approval that has been done, gives a certain amount of confidence in it, we hope. So with that in mind, I'm going to move on to part 2. And this is the why we're all here. So now I'm going to talk about some of the discoveries that have been published in nature journals, science journals, various places, but just giving us a little bit of a sort of a roundup of some of these latest discoveries. So some of you may have seen this image a lot when you were growing up, maybe at school, and it's called the March of Progress. So it's what I'd call maybe an outdated way of viewing the progress of human evolution. So in it, you've got one step after another, that this kind of ape-like thing leads to a bit more of an upright ape, leads to progressively, and then we have very much the evolution of man, not woman, at the end, just this big strong guy. And we now know that that's pretty outdated. Some of you may be more used to seeing this sort of tree of life image, where we still have kind of single species here, but we're not suggesting that one led to another, because they're all different branches along this evolutionary tree. You can see that we're all related because we all have this trunk connecting us, but that we are not descended from apes. So this kind of tree of life metaphor is, has been sort of classic throughout kind of the study of evolution for quite a long time. But these days in human evolution, we've moved to what we think of more like a bush than a tree. So in this image, which I think is still actually, it's an acacia tree, but it's a bushy tree, you can see that there are lots of parallel branches. So these are all, apart from us up here, these are all extinct species. But we can see from this that there were multiple species alive at one time. So we know, if you think about the world today, most species, there are different kinds of species of, say, horse. There are different horse subspecies and species. It's kind of rare that only one species of a particular lineage would be alive at the time. And that is the same way with our human ancestry. So we see this kind of bushy nature of human evolution, and we see how many of these different hominins were around. And we have different branches. So we've got the Homo group, the hominins that I was talking about, who are our closest relatives. We have the Australopithecines, who you may also have heard of. And one of the reasons that say that Homo naledi claim about all those sort of sophisticated behaviors is controversial is because some people think that actually Homo naledi maybe has more in common with Australopithecus than with Homo. And we don't know of any of those kind of sophisticated behaviors, certainly in any of the Australopithecus group. There's also another relative, the Paranthropus group, the Ardipithecus, and this is going back as far as we know. These are the oldest in the hominid lineage. Going back 6 million years ago, we know about Sahelanthropus and Auroran. And the amount of which we know about each successively older species is vanishingly little because of the amount that remains today. But because we're talking about whole 6 million years here, and I can't possibly talk about that in the something like 24 minutes that are remaining, I'm just going to zoom in on this top branch here. And to tell you that even though, amazingly, though it looks like this bushy thing is hopelessly busy and there's so much going on in it, this is outdated. You see this million-year period here. We've only got 1, 2, 3, 4 different groups alive here. The thing is, these days, we know there's a lot more. So there are 1, 2, 3, 4, 5, 6, 7, 8 different species of Homo, that's the H here, within that 1 million year period from then to today. And so we've got us here, and this represents the span of our species. So this is about 300,000 years long, and I'll come back to that in a second, but you can see the overlap here. So we have this species overlap, this one, this one, this one, this one, this one, and this one, which is kind of cool because we now know that actually going back to that idea of the progress, the one leads to another, that is not at all the case, that these contemporary species, we did interrelate to them, we interbred with them in certain cases. That this is all happening at the same time within the last 1 million years. So I'm going to talk a little bit about how we know that. And to do that, I'm just going to talk about quickly a method that is really revolutionizing paleoanthropology, human evolutionary studies, or rather 2 methods grouped together as what they call molecular techniques. And first of all, to give you an analogy with how that is really breaking so much ground in the field, I want to go back to the 1950s. I really like jumping back, 50 years, 150 years to give you the sort of start context to all of this. But in about 1950, radiocarbon dating as a technique came into archaeology. And I don't know how many of you know how that works, but basically, every living organism absorbs a certain amount of carbon from the atmosphere. And when the organism dies, the amount of carbon within them breaks down. And because it's a radioactive decay, you can measure the proportion of carbon that remains in something, and you can work out from that how old it is, how long since it was last alive. And this works as far back as something that's 50,000 years old, because once it's 50,000 years old, the amount of carbon-14 that's left in it will be so small as to be basically not measurable. And the thing that radiocarbon dating did for archaeology was that it basically freed up brain space. So archaeologists till that point had spent so much of their time trying to work how old everything was, because if you don't have a direct method of dating a piece of bone or a piece of leather or something like that, you then have to work out how old it is by relation to other stuff, which maybe you can tell because things like trees, put on serial rings every year, and you can date that a bit. So you can try and relate that to the thing you really want to date, but it's a really tricky process and there's a lot of error in it. So once archaeologists had a method by which they could directly date things, they could say that this fossil is 34,000 years old. They could then turn back to the more interesting questions of why it was there, how it was there, what did it mean? And the same kind of thing is happening in human evolutionary studies. So going back to all of these guys, whereas maybe 25 years ago, 30 years ago, we would have been spending a lot of brain effort in thinking, well, how did they relate to each other? Who's more closely related to whom? Who's the sister on the metaphorical tree as opposed to, say, the cousin? These techniques are answering that and meaning that we can free up brain space again. So the 2 techniques I want to talk about quickly are ancient DNA and ancient proteins. So I hope this is visible, but I'm going to go through it a little bit, even if you can't read all the things. So we have ancient DNA and we have ancient proteins. And a bit like the carbon I was talking about in radiocarbon dating, these survive to a certain extent in organisms after death in the ground where they're excavated from. So up here you can see that the little symbol on the left is DNA, the little symbol on the right is protein, and where it's worked, it's in green, and where it hasn't worked, it's in red, and where it's in gray, it wasn't tried. So we have been able to extract ancient DNA from a Denisovan tooth that's 30,000 to 50,000 years old. Also ancient DNA, ancient protein from a 90,000-year-old Denisovan. But on the other hand, Homo floresiensis, which is a bit younger than that, neither DNA nor protein seems to have survived. And that's because the places where they are buried is really important. So in a tropical environment, these molecules don't really preserve, and they certainly don't preserve as long. So we go back to a hot and dry environment, 3.8 million years ago, we have protein surviving, and that's about the oldest protein that we know has survived. And why is this informative? So ancient proteins are very useful because they can give you a sort of a loose idea of what something is. So while they won't tell me if something is Homo sapiens or a Neanderthal, they will tell me that it's Homo, that they're a hominin relative. Ancient DNA, on the other hand, is a much more in-depth technique where you can get an entire genome. You can learn about these things like who was interbreeding with who, and I've got a really exciting story about that in a minute. But I just want to talk again a little bit about the preservation of these things because it really dictates what we know and where we know it. So while we don't know a lot about stuff that's preserved in tropical environments going back this long, this picture here is what an area of Greenland would have looked like 2 million years ago. And we know that because the preservation within permafrost of ancient DNA is so good that they've been able to recreate an entire ancient ecosystem, which is incredibly exciting. Now, I was talking about ancient proteins and ancient DNA. The great thing is that these can work together. So this is a tiny chip of bone that is, I think, probably about the size of my top finger knuckle joint here, which is incredibly uninformative to the naked eye. So you would get this out of the ground. Not only could I tell you if it was, horse or human, I couldn't tell you what part of the body it came from. I have absolutely no idea. But the thing about the protein, the ancient protein analysis, is that another thing it massively has in its favor is that it's cheap. So there's a method by which you can screen thousands of these tiny little chips. And while it won't give me detailed information on what species it comes from, I can at least tell that kind of broader grouping, that it's from, say, the Homo group, or it's from, a lion or a tiger or something. So from this, and among thousands of other little tiny chips of bones excavated in a Siberian cave, they ran it through the protein analysis and found out, oh, wow, this is hominin, this is human, this is really, really exciting. We're going to now spend our effort on doing ancient DNA, which is a much more expensive technique. It's a much more time-consuming technique that you couldn't possibly use on the thousands of tiny bone chips that you might dig up in a cave. But you can do it on this one because you already know it's special. And this is one of the most special tiny bone chips in the entire world, because we know from this that this girl, she was a girl, had a Neanderthal mother and a Denisovan father. I'm going to tell you a bit more about Denisovans and Neanderthals in a second. But we know that this is an example of two human species hybridizing together. We know that they could interbreed, that they could live together. They obviously were not in conflict all the time. They were living in some kind of harmony. And that's really exciting. And I did mention that, mostly it's the ancient DNA that's really exciting, but at the moment there's a really exciting paper coming out with protein data from a 2 million-year-old fossil, which again is really, really old for this to preserve, but it's going to be able to tell us about the sex of the different fossils. So we're starting to be able to tell all of these sort of basic biological relationships from these techniques, which is allowing us back more brain space to try and think more about their behavior and things like that. So I mentioned those, we saw them on that timeline, all of those 8 species that maybe overlapped. And now I want to talk about the possibilities that our species, Homo sapiens, might have encountered them And like the Neanderthal and the Denisovan hybrid that we might have hybridized with other species while we've been wandering around the planet. So this is, at the moment, we think, the oldest Homo sapiens fossil from a site in Morocco called Jebel Irhoud. And the, sorry, Ka is 1,000 years, so K, 1,000, A, annum. And from this place, and actually it's really all of Africa, because although I've just marked this as a find spot, there were probably Homo sapiens populations all over here. And you can see this arrow is starting there for that reason. And these arrows then mark the way that our species spread out of Africa all around the planet. And some of the years marked on there tell us about fossil find spots in those areas, or maybe stone tools that have been found that help us to date those progressions. So we know that starting out here, we have our species spreading into Europe by 43,000 years ago. Spreading out, you can see that some of the dates are kind of funny. So we know that, you know, you've got 32,000 years old there. So I can't actually tell you why that's an earlier date than that one, other than that people may have moved. This is only representing a linear progression, whereas actually there might have been a lot of back and forth, up and down, spreading out, spreading in. But we know that they're getting down to Australia, 65,000 years old at a time. And I'm afraid I don't have the Americas on this map because this is a much more recent progression of our species. And I'm just talking about these sort of old world meetings. Now, those are all those 8 species that we had on that timeline again, and this is where they've been found. So the 2 I'm not going to talk much more about this evening. We've already had Homo naledi, and the reason that I said that that's a possible candidate for something that our species might have met is we know that Homo naledi was alive was alive around the time that we first evolved. So it is possible that we could have met them. Homo heidelbergensis is another one that's around. Homo heidelbergensis, though, suffers from the problem of it's kind of before that ancient DNA limit, or at the end of that limit. So it's kind of about 500,000 years old, and that's when ancient DNA stops working, that it just tends not to preserve anymore. So this, how we relate to Homo heidelbergensis through our molecules is kind of an open question, and it's maybe going to be the next big one to answer now that we've cleared up some of these later relationships with other human species. So I'll leave these 2 for now, but just to say that heidelbergensis is probably the candidate for the most likely joint ancestor of us and Neanderthals. That's maybe the last time that we shared an ancestor. So you can see from this map that we know that there's been genetic admixture, that means interbreeding, that we've shared DNA with Neanderthals at these red circle spots in these areas. And we know that there was admixture with Denisovans at these ones. So these are the ones we know about. We know that our species definitely met these guys. But what about these other 3 species that I had on that slide before? We were spreading out down here around this time, so might we have bumped into these guys as well? I'm going to talk a little bit more about what we know and what we don't know there. So just to talk quickly about the ones we know about. So we've got some more of these sort of dating profiles here. So we know that Neanderthals certainly overlapped with us. They overlapped with us both in kind of date range as well as our regional range, Eurasia being Europe and Asia. We know that Neanderthals were amazingly well adapted to cold climates. They had lots of bodily physiological adaptations that meant that they did very well in the cold. They had bigger bodies, they had bigger noses that would have made them sort of breathe warmer air more efficiently. But we also know about their behavior, that they built weird ornaments out of stalactites. They had rock art, they had like ornaments, maybe jewelry. We also know that they really liked interbreeding with other hominins. We know that Neanderthals, who is these guys in red here, this is another one of those tree diagrams. We know that they not only interbred with us, where it says early modern human lineage, that's Homo sapiens, but they also interbred with Denisovans and maybe someone else who I'm going to come back to. What do we know about their dislikes? Maybe they didn't like warming climates. Maybe those things that meant they were so well adapted to the cold didn't do them so well when it was getting warmer. Maybe they competed with us. We don't have very much evidence when it comes to, warfare, any kind of direct competition like that. But populations might have expanded at different rates, meaning that they eventually just disappeared. They dwindled. We've also got Denisovans, who are the really enigmatic ones. So I'm just going to go back a second. So these little bone chips and this jawbone, those are like the biggest fossil remains we have from Denisovans. We have so little skeletal material from Denisovans that we don't even have a species name for them. They're called Denisovans, whereas everyone else gets a Homo name. See, so they've got this Linnaean binomial. So Homo is a bit like the surname. That shows that they belong to the human group. And then they have these species names, like a first name, except it's the other way around. Denisovans don't get that because you need enough skeletal material to be able to designate a new type for a species. That's how taxonomy, the science of naming new species, has always worked, that it's done on morphology, bits of body. But we don't have enough of that for Denisovans to do it. So it's crazy that we know huge amounts about their genetics. We know about how Denisovan genes that have introgressed have come into our DNA sequences as modern humans, how those can confer better resistance to certain forms of diseases. We know crazy amounts about their genetics, but so little about what they look like or what they did. We know very, very little about their behavior. This is basically it. That's really all we know about them, except that they did interbreed with us and they interbred with Neanderthals. And now I'm turning to those other three enigmatic ones. That this is, if you take that first map I had with the sort of arrows spreads out, this is what it looks like for islands, Southeast Asia and Australia. So this is where we got to Australia, our species, 65,000 years ago, and we probably took this route. So for this map, all you really need to see are those orange arrows there are showing the movement of our species throughout these islands. This kind of island hopping thing that we would have had to do to reach Australia. But I mentioned a few slides back when I was talking about the preservation in tropical areas. It's really bad because stuff just doesn't preserve, it decays. So we have very little comparative fossil evidence for this region than we do for Europe, where it was cold and glacial, and we have things like that permafrost I showed in Greenland. That those were the kind of conditions in which things like DNA and protein survived really, really well. But we do have some bones. So we have Homo erectus here. We have Homo floresiensis here. And we have Homo luzonensis here. And here's their little dating profiles. So I'll come back to that abomination on the right in a second. For the start, we've got Homo erectus. Now, Homo erectus are amazing. Look at that date range. That is crazy. This is the longest lived hominin species by far. So if you think about the first appearance of our species, Homo sapiens, 300,000 years ago, we haven't been around for very long at all. Whereas these guys lived for well over 1 million years. And these were the first guys to be those kind of cosmopolitan globetrotters. They left Africa, they went to Europe, they went to Asia. They made it as far, they nearly made it to Australia from Africa. That's a huge spread. They clearly liked travel. They were bipeds. They were really, really efficient runners and walkers. We also know that they may have had ornaments. They had stone tools, like hand axes. If you've ever seen a beautiful sort of teardrop shaped stone tool called a hand axe, that is very much associated with Homo erectus. And then these guys are even more enigmatic still. So Homo floresiensis, you may have heard of the Hobbit. It's because it was this very, very short little guy. It's been nicknamed the Hobbit, and it's found only in one set of islands in Indonesia. And there were these tiny little diminutive hominins, and they were associated with really diminutive animals as well. So pygmy elephants and Komodo dragons, and they seem to have been really good at hunting them. But that might also have contributed to how they eventually went extinct, because as they ran out of resources on the island, maybe their body size is reduced, but eventually you exhaust all the resources there, and unfortunately you may go extinct. We also have Homo luzonensis, and this is maybe competing with the Denisovans to be the most enigmatic fossil species of hominin, because all we have for luzonensis is 5 teeth, which are enough to tell us that it's a separate species. And crucially, Homo luzonensis gets that nice extra bit of kind of kudos in the form of a full species name, because you can designate a species based on teeth, but you can't do it based on genomes. This is the most recent arrival to the hominin group. That this, I think, was published maybe in, I'm going to say 2019 was when this discovery was published in Nature. we know very little. We know that they may have a pretty long range. We know that they're found in the Philippines. We know that they're found with rhinos. Some people say it might actually just be a form of Homo erectus. So that's their dislike, is they want to be their own species. It seems pretty likely, because of the overlap in range and the overlap in dates, that Homo erectus probably had these kind of offshoots, that these two species maybe arose from a Homo erectus lineage over time. But it's kind of speculation based on the morphology. But just going back to this chart where I showed, all you need to know from this is the Neanderthals interbreed with us, and they also interbreed with Denisovans, and we interbreed with Denisovans and with Neanderthals. But we've also got this mystery hominin. This is very excitingly called something called a ghost lineage. It's when we find bits of DNA that we know don't come from these species. It must come from a different hominin species, but we don't know what it is because all we have are genetic fragments. It's like you've got footprints, but you've got no idea what the footprints are from. But because of where we know that these people were found, I'm very happy calling all of these species people, they certainly were. Denisovans are found in Eastern Asia. And so we're looking for a hominin candidate probably in Eastern Asia, which means it most likely, well, you'd think, is one of the candidates would be one of these guys for the interbreeding into Denisovans. That would explain. One of these is our mystery hominin. But at the moment, we haven't got any evidence for it. So we have got the issue that the genetic molecules, the molecular techniques don't really work so well in these tropical areas. But what you can use are computer methods to try and model the amount of admixture. So this is a study that we published a couple of years ago, trying to work out what that ghost lineage might have been. And all I can tell you from this is that our best guess, based on the evidence we currently have, is that none of these guys are the mystery hominin. And what does that mean? That probably that bush isn't yet bushy enough, that there are more hominin species out there to be discovered. And as our techniques improve, as we find out more, as we excavate more areas of the world, we are probably going to find more and more hominin species. And I just wanted to talk quickly, I'm really wrapping up now in a second, about some of the other things, but I've talked about the genomes and the bones, but not so much about the behavior. So what I really hope is that as we have these new, really exciting scientific techniques revolutionizing what we know, we can answer those questions that plague us, the really basic questions of who's related to who and that kind of thing, is that we can free up more space to do my favorite thing, which is to think about how people behaved, how we interacted. And all of these behaviors that are depicted on this slide are what you might call symbolic behaviors, things that relate to not just sort of basic things like eating, feeding yourself, covering your kind of basic human needs, but speaking to something beyond that, this kind of symbolic interest that maybe is something that's characteristic of our human group. So this is an incised and engraved toe bone from a giant deer, and it's definitely in a Neanderthal context. We know that Neanderthals were carving things to use as jewelry. These are teeth and shell ornaments that would have been worn maybe as a necklace or something like that. Also in Neanderthal sites. I mentioned earlier these sort of stalactite and stalagmite structures. This is an underground cave where Neanderthals, for some reason, were making these kind of crazy circles with sorry, getting overexcited about that. Maybe they were getting overexcited about the stalactites too. They were throwing them all over the cave, making beautiful structures. And then we have, apart from the Neanderthal examples of this, we've got even older ones. So I mentioned that Homo erectus, that's that really long-lived species in Southeast Asia, spreading out from Africa, who is much older than most of these other ones. But we know that they were engraving shells. This is the shell from Java that has this sort of crosshatch pattern engraved on it, which is crazy to think that they evidently had some kind of abstract meaning associated with this. And then just going back to Homo naledi briefly, I think it certainly is possible that hominidi engaged in some of these symbolic behaviors. I have absolutely no problem with the idea that they would have had the sort of the cognition, the brain capacity to do these things. It's just that we don't yet have good evidence for it. We may absolutely find that evidence out in the future, and I really, really hope we will, because it's a bit like what people say about if we're the only if we're the only ones alive in the solar system, in the universe, kind of a waste of space. You know, it's kind of a waste of the world, or of all of these different human species, if we can't allow them the idea that they could have had these symbolic behaviors where the evidence exists and it's well supported. Let's absolutely believe it. It's so exciting and really cool. But we do need to have, I think, a pretty high standard for the evidence basis to believe this, because it's really important. And I'm going to leave it there. This is me with another hominin relative doing my best to imitate it. And if you're interested in any of this, you can check out Nature and Nature, Ecology and Evolution at these websites. If you're interested in just kind of reading news or commentary on these things, and that's written usually in a pretty accessible way, there's a really, really nice newsletter that you can get from the Nature journals that tells you about these really exciting developments in quite an accessible way, and it's a good thing to sign up for. So thank you so much for listening. I hope it was coherent, and thank you for coming out.